I'm going to actually talk what I would call unconventional clinical trial designs, and have been asked to talk about phase 0, umbrella, and basket clinical trials. And as you can see, I have no disclosures. I think for many who are listening in, this is probably old news, but what are the drug treatment challenges in primary brain tumors, particularly glioblastoma? Of course, one of the big ones is drug delivery, and that's our interest in the phase 0 concept. Everybody knows about the disease heterogeneity, and we got really one of our best snapshots several years ago, work from Mario Suva and Brad Bernstein looking at single cell RNA sequencing and recognizing that even in a small geographic area, there is quite a lot of diversity of the tumor cells, very important when you think about targeted agents. Of course, the tumor itself is heterogeneous. When you just look at it, there's a central necrotic region, the leading edge, which can be different, and infiltrating tumor in the surrounding brain, which turns out to be one of our biggest challenges in treatment. And of course, these tumors can become resistant to our therapies quite quickly. And then again, from the clinical perspective, and this certainly impacts clinical trials, differentiating progression from pseudoprogression, sort of the classic example of the post-chemoradiation scan that gets spontaneously better, and then in reverse, response and pseudo-response. And here's an example of a bevacizumab pseudo-response scan gets better, but not a lot of change in the tumor volume. So since we are going to focus on conventional clinical trials, I thought it would be important to talk about unconventional trials. We'll talk about conventional trials. Of course, everybody knows the phase 1 studies designed to determine the maximum tolerated, or in the case of biologic agents, the optimal biologic dose. And contrary to sort of the current trend, really not designed to determine an early efficacy signal. By definition, most of the patients on a phase 1 study are not getting the optimal dose. They're either getting a lower dose or a dose that was too high and toxic. So really the signal should be reserved for the phase 2 trial, which is the first true test of efficacy. And there have been iterations of this 2-stage designs. In case an agent really doesn't work, you don't want to expose that many patients. And then, of course, the use of a randomized phase 2, and we don't have time to talk about the nuances there, but hopefully we see a signal, and then it goes to the phase 3, which is really the definitive randomized trial, often accompanied with a placebo, so a double-blinded phase 3 design, the best. So we are focusing here on the phase 0 paradigm. I wanted to point out what I consider to be the seminal paper published by Murgo and colleagues, including James Doroshow, who really pioneered this work. This is in Clinical Cancer Research in 2008, and that little snapshot is, I think, one of the key figures. And what it emphasizes is that it's really important to do appropriate preclinical testing before you embark on a phase 0 trial. So I think in the context of neuro-oncology, brain tumors, to really pick the models that most closely recapitulate the type of treatment that you're looking at. So if you are focusing, for example, on an EGFR inhibitor and want to look at pharmacodynamics, you want to have a spectrum of models, some with amplified EGFR, others without, so you can do a comparison and really test that your bioassays are accurate. And then you're going to go on to confirm this in clinical trials. And again, in the preclinical model, you get an early sense of delivery. You could validate your assays of PK and BD and pharmacodynamics, so pharmacokinetics, looking at drug delivery, pharmacodynamics, looking at the effect on the target. So really, really important. And I think that's underscored in this study that was actually published in breast cancer, where they asked a question. They had an inhibitor of AKT pathway, ERK pathway, And they wanted to ask the question, in a sample obtained without treatment, is there a difference in their biomarkers before they do the phase 0 study in time? So they had two groups. They would have a quick section removed from the breast cancer and very quickly processed, preserved, and then the actual resection, so the lumpectomy. And as you can see on the left-hand side, the estrogen receptor, proliferative index, HER2, remained quite stable between the two, but the phospho species of their targeted pathway, phospho-AKT, phospho-ERK1/2, there was a very big difference whether there was at first a quick section reserved or then the actual removal of the entire tumor. And so had their study included their agent, their targeted agent, and they only looked at the subsequent mastectomy, they would have obviously subsequent removal falsely felt like they had actually inhibited their target, when in point of fact, that was just the spontaneous loss of the phosphoepitopes, phospho species of these proteins. So really important, the take-home message being you have to establish all the potential confounding issues before embarking on the clinical trial. So I've talked about phase 0, but in point of fact, if you look at what the phase 0 definition is, they think it should be performed before the phase 1 study. And in fact, the FDA has a special category called an experimental IND. You use lower than therapeutic dose, so you essentially eliminate the chance of any toxicity, and you're really focusing on pharmacodynamics. And again, this is designed for systemic cancers where you don't really worry about delivery. And so you really want to look at whether you're hitting your target before you embark on more studies. In the neuro-oncology space, we're really talking about what we call window of opportunity. And here, you really need to do the study after the phase 2 dose is established. So you do the phase 1 study first, and you get your phase 2 dose. So you're using the optimal dose. And here, because of the blood-brain barrier, most of our studies are focused on drug delivery. And as you will see for CNS tumors, we often recommend that not only the central part of the tumor where there's contrast enhancement is sampled, but also other areas which may have very, very different pharmacologic properties. So what are the paradigms that one can use for these windows of opportunity studies? At top, talk about the biopsy, treat, biopsy, or biopsy, treat, resect. That's optimal, of course, for pharmacodynamics. So you have a baseline of what your target looks like. You then treat the patient, treat the tumor, take it out, and then see if you've hit your target. Of course, you can use the resected tissue to also evaluate delivery. This is very difficult because it's often hard to get coverage from third-party payers for 2 procedures in a patient. The second design is a random allocation. So one group of patients gets the treatment, the other does not, and you see whether the target is more impacted, or a higher percentage of patients have lower target with treatment than the control group. And this was actually used by Dr. Friedman and colleagues when they looked at 6-benzylguanine and showed that the control had high levels of MGMT in most of the tumors, whereas the patients treated with 6-benzylguanine, there was no MGMT, and they determined, in fact, that it was effective. And then you can actually do the treat and biopsy or treat and sample and compare a tumor from the tissue bank as a historical control, but you have to do very careful statistics there. So again, one of the caveats here is the optimal time to measure delivery, the pharmacokinetics, usually at the Cmax in the blood, may not coincide with the optimal pharmacodynamics, particularly if there's a lag from the time the drug gets to the target to has the downstream effect. So that should all be modeled preclinically. So again, the eEANO group put together phase 0 recommendations that were published in 2020. And again, a patient must be undergoing a planned tumor resection. So we don't do this really only in patients who don't need an operation. Use the clinical dose of drug. Again, that's the phase 2 dose. And they do recommend, again, not only sampling the enhancing part, but also trying to get tumor adjacent regions, non-enhancing tumor, et cetera. And then try to do correlative studies looking at cell viability, proliferation, and again, the target of the drug, that's the pharmacodynamics. So I have a couple of examples. I was fortunate to lead a phase 0 or window of opportunity study of selengitide. We used 2 doses, 500 milligrams. The patients received 3 doses before they went to the OR, or 2,000 milligrams. And what I want to point out is at the time of resection, there was actually more drug in the tumor tissue, which is on the right-hand side, than there was in the blood. And so this was, I think, confirmation that we had not only good delivery, but actual retention in the tumor. So with this information, selengitide was tested in a phase 3 trial. Unfortunately, it was negative, but we at least knew that this was not a drug delivery issue. We have the example of lapatinib, which is a dual HER2 EGFR inhibitor. And this was tested in patients with recurrent EGFR amplified or EGFR V3 mutated glioblastoma. You can see recurrent disease. The patients received lapatinib for a week before they went to surgery, and then continued on the lapatinib. This was an NABTC trial. And they knew from preclinical work what concentration of lapatinib was really needed in the tissue in order to really see an effect. And you can see the line, that dark red line in the middle, and very few of the samples actually had sufficient concentration to be likely effective. And when they did the correlative studies, that's the right lower hand corner, you can see that there are lower levels of EGFR activity that corresponds with lapatinib treatment, but there still is quite a bit of EGFR. So there was not a sufficient blockade to affect clinical or impact clinical efficacy. Again, very helpful window of opportunity study that should guide future dosing of lapatinib. I would be remiss if I did not talk about microdialysis as a way to measure intracranial, intratumoral concentrations. This is a small catheter that gets inserted into the tumor. It's minimally invasive. It uses passive diffusion, and it's been used routinely in evaluating traumatic brain injury. And in fact, patients have been known to walk around with the catheters for a week or more. I show an illustration of what it looks like and implanted in the brain and the device, and a little bit of animation showing that with fluid going through the microdialysis catheter, it forms an equilibrium with the extracellular fluid, and you can actually calculate over several days what the concentrations of drug are. Not all agents are amenable to microdialysis. There's a limit on the molecule size, and molecules that tend to be sticky don't really fare well. They wind up sticking to the walls of the catheter. But in the appropriate circumstance, microdialysis is a good way of measuring tissue pharmacology. All right, so to finish up on phase 0 studies, the first question in Neuro Oncology is, where do these studies belong in the drug development plan? Do you do it early on? And here the big question is a safe dose established? And what about the pharmacokinetics? So when should the sample, the tumor be sampled? Right, so you want to get it at the Cmax, so you're getting your maximum concentration. But again, the problem is that, do you actually have the optimal dose? In Neuro Oncology, it's recommended it's done late, so we have the optimal dose. We don't use a subtherapeutic dose. But after you've done all the work in a phase 1, you do the window of opportunity study, and you discover you don't get good delivery, so all of that effort really has gone for naught. So it's a dilemma, but as of now, the recommendation is to do it after the phase 1 or phase 2 dose is established. Again, when to sample the tumor, the PK best at Cmax, but the PD may be later, and you should know this from preclinical testing. So what are the pitfalls? Of course, when a tumor is removed, it is contaminated with blood, and blood concentration of the drug will be part of the assay. So when you do see tumor concentrations near or exceeding blood levels that are done simultaneously, that's a pretty good indicator of good delivery, like we saw with selengitide. And again, the caveat to know the characteristics of the pharmacodynamic markers, as I showed you the example of phosphoproteins that are quite unstable. So if you wait too long, you'll get a false indication of effect. Now turning now to novel clinical trial designs, the umbrella and the basket design. The umbrella is testing the impact of different drugs, different mutations in a single type of cancer. And the basket is you're going after a target across many different types of cancer. All have a target strategy. The underlying premise of these strategies is that cancers have an oncogene addiction. And as I mentioned, those little cartoons of the umbrella with a single type of cancer and the basket, multiple types. So starting first with the I-SPY2, which was in patients with newly diagnosed breast cancer, it used an adaptive design. Patients were grouped by hormone receptor, and they wanted to see, whether they could target the molecular signature. Of course, they had to stratify by the presence or absence of HER2 expression. This included interim biopsies, and an endpoint was a pathologic complete response that was verified with resection. But importantly, if at the time of surgery they did find a residual tumor, they were able to adapt. And so this was an iterative process, and the successful strategies continued on, and others were dropped off in the adaptation process. The Shiva study was looking at progression-free survival, a variety of targets, which you can see in the table in the upper right-hand corner. And they were targeting 3 pathways: hormone receptor, PI3 kinase pathway, or RAF-MEK pathway with a built-in crossover. And so this was a very complicated and a robust schema. They really left nothing to chance and highlighted everything in their algorithm to make sure that this was done appropriately. But they really focused in on this idea of the randomization when there was a molecular targeted agent. So patients were then either sent to the conventional therapy, oncologist choice, or the molecular targeted therapy. And if the patient failed on one, they could cross over to the other. And importantly, there was a report that showed that they had randomized 194 patients, and the survival curve, or the progression-free survival curve, showed no difference with that primary choice. So an elegant study, but unfortunately, it did give us an algorithm, but not an efficacy signal. So we have additional designs like the umbrella design, which is a multi-arm, multi-stage design. You can see here in the figure on the left-hand side, the traditional approach. You do a phase 2 study that leads to a phase 3 study. You have a control arm and your phase 3, your control arm, your phase 3, et cetera, et cetera. The multi-arm, multi-stage, single control arm, multiple arms that if they underperform can drop out. And so this was used in the STAMPEDE trial for prostate cancer. You can see the top line is the standard of care that continues on, and then multiple experimental arms. The early ones all dropped out for lack of efficacy, and the design allowed them to roll additional arms. And then colorectal cancer, the Focus 4 now gave patients standard chemotherapy and then by molecular selection, a targeted agent, and then they were subsequently randomized to either placebo or other targeted therapies to see whether continuing targeted therapies in the maintenance state would help. So again, the same type of multi-arm design. NCI Match, quite a famous basket trial, now looking across cancers for specific molecular target. Single arm design, looking at mutations, fusions, translocations, amplifications. It was rule-based. The amplification testing was defined very carefully. And they used the most recent tumor specimen, which is in fact quite important. And there was an opportunity for patients to actually get additional therapy if they failed the initial. So we do have results. Remarkably, nearly 6,000 specimens were tested. They had a 93% technical success in doing the molecular and immunohistochemical analysis, and they found that 37% had an actionable alteration, and they were able to assign treatment in just 18%. And 11% actually had multiple actionable alterations. And importantly, they did discover that over 70% had a resistance-associated alteration. So again, a small percentage of patients across a variety of cancers actually had a targetable opportunity, but very important to actually develop the logistics with that high degree of not only accrual, but technical success. In the neuro-oncology world, we have a couple of great examples. The INSIGHT study being led by colleagues in Boston. Again, this is an umbrella trial, newly diagnosed, glioblastoma, MGMT unmethylated, so they didn't need to, in the experimental arms, worry about the addition of temozolomide. And they did it as biomarker agnostic. And you can see that it does have an adaptive design. And they also included this concept of dropping arms that are underperform and adding additional arms. And the goal here is to actually look for signal that would then lead to a definitive phase 3 trial. And then the other big study in neuro-oncology is GBM Agile, which has multiple components using biomarkers, adaptive design in both a newly diagnosed as well as recurrent glioblastoma. And finally, in the CNS space, our program at the NIH for rare cancers called NCI Connect. And here we have a basket design for 11 different rare cancers listed there, and they get the checkpoint inhibitor nivolumab. We actually have made this a multi-center study across the United States, 10 additional centers. And we've stratified patients actually by number of prior therapies, which seems to be more predictive than the actual cancer type. And I'm happy to report at this past SNO meeting, there was a preliminary report in patients who were heavily pretreated by the presentation by Dr. Marta Penas-Prado, who's the PI of the study, and who actually received a SNO award. So let me finish up with what I think some of the future may be, and this is amenable to both the umbrella and basket trial, and this is synthetic lethality. I show an example, classic example of synthetic lethality. Tumor cells with a BRCA mutation rely on PARP for DNA repair. And if you use a PARP inhibitor in a normal cell, BRCA helps DNA repair. If you use a PARP inhibitor in a cell with BRCA mutation, like a breast cancer, it leads to cell death. That's the concept of synthetic lethality. Again, in wild type cells, you have two genes that cover for each other, and you target one of the genes, the cell's perfectly happy. In a cancer cell where there's a deficiency, mutation, deletion, you target its synthetic lethal partner and it cannot live. So here's a nice overview. I'm part of a group here at the NIH led by Eitan Rupin and with very close collaboration of Ken Aldape, looking at synthetic lethality, and they call it a SELECT. And so in vitro data using drug response and gene knockout using CRISPR and shRNA, tumor data, so tumors with a lot of synthetic lethal partners do better, and actually phylogenetic data, because these tend to be inherited. So ones that are phylogenetically, genes that are phylogenetically related are more likely to be synthetic lethal partners. You develop a network, those targets that have the most interactions with other genes are most likely to be beneficial, and you can actually do response prediction and then stratify and hopefully find agents that are good for that patient and their tumor. So here we can do adaptive testing. So you take the top priority from your synthetic lethal evaluation, you do target specific agent. If you see a response, you continue treatment, and in adaptive design, this gets a high priority. If there's no response, it gets a lower priority, and that adaptive design is used in the context of synthetic lethality to find actually the best agents, very similar to Stampede. And then once you're at a point where you think you have synthetic lethal agents, how do you determine if they're beneficial? Well, to focus in, you randomize between the specific synthetic lethal targeted therapy versus standard salvage, and see whether you actually outperform what you would have done in typical circumstance. And of course, opportunity for patients on the standard arm to switch over. So let me just summarize. We have targeted therapy using basket and umbrella clinical trials across cancers. There's been modest efficacy, but I think really a lot of work helping us in the logistics and design. And of course, we, particularly in Neuro Oncology, have to define the endpoint. eEANO's been working on this progression-free survival, overall survival, response rate. And of course, the question, can these strategies be improved? Of course, the answer is yes. And I would like to leave you with the idea that approaches like synthetic lethality may in fact help us, at least in the sense of targeting specific pathways. And with that, I will close and thank you for this great opportunity.